Hello, student. This is our second last lesson in transport. Very interesting. We want to talk about blood groups. Do you know your blood group as an individual? So the human beings have what we call the ABO blood group system. And there are 4 types of blood groups in human beings. That is blood group A, blood group B, blood group A, B, and O. And these are based on types of proteins called antigens present on the cell membrane of the red blood cells. And there are 2 types of these antigens on the red blood cells, denoted by letters A and B. In the plasma are antibodies specific to these antigens, denoted in small letters A and B. So remember, the antigens are present on the cell membrane of the red blood cell, and they are denoted using capital letters. The plasma, the antibodies corresponding to these antigens are denoted in small letters, and they are located in the plasma. Very important. If somebody asks you the difference between antibodies and antigens, we shall learn this in our next lesson, but briefly, antigens are just like foreign materials in the body, but antibodies are produced by the body to defend itself against antigens. So a person of blood group A has A antigens on the red blood cells, and the corresponding antibody is B. Remember, the letter is small one. A person of blood group B has B antigens on the red blood cells and A antibodies in the plasma. A person of blood group AB has A and B antigens on red blood cells, and therefore no antibodies. And why do you think is the reason? We shall answer that later. A person of blood group O has no antigens on the red blood cells, but will have A and B antibodies in the plasma. Individuals with antigen A in their red blood cells will never have antibody A. That is, but will have antibody B in their plasma. So this person having antigen A is having blood group A. So whenever you see this antigen A, that means this is a blood group, and so this person cannot have antibody A, but will have antibody B. Reason we are answering: agglutination, or what we call clumping together of the red blood cells, would take place if the antibodies corresponding to the antigens come in contact. That means the blood will clot in the body and will block, therefore no blood flow, and that is likely to lead to death. So let us look at this table where we are looking at the blood groups, the antigens on the red blood cells, and antibodies in the plasma, to summarize what we have just discussed. So these are the blood groups, and we've said we'll have antibody or antigen A, but we'll have antibody B, not A. Blood group B will have antigen A, antibody A in small letters. AB blood group have antibodies A and B, but no antigens. That is, we said that is to avoid the clumping together of the red blood cells. Blood group O has no antigens, but will have both antibody A and the B. We want to look at blood transfusion. So this is the transfer of blood from the donor to the circulatory system of the recipient. So normally there are those organizations like the Red Cross, which carry out this blood transfusion, whereby to assist those people who have shortage of blood, maybe in women during delivery, a woman has lost excess blood, or in case of accidents, or it's just some sicknesses, and the individual is found to have little blood. So this knowledge of blood group is essential at this time of blood transfusion, because we are going to see how. So a recipient will receive blood from a donor if the recipient has no corresponding antibodies to the donor's antigen. If the donor's blood and the recipient's blood are not compatible, that is, agglutination occurs whereby the red blood cells will clump together, and that is likely to lead to death. So when we say compatible, that means they do not agree because of the of these antigens and antibody reaction. So look at this table. This is the recipient, or the person going to receive the blood, and this is the donor, the one giving out blood. So this person, the recipient, is having blood group A. That means the antigen is A. Of course, the antibody will be B. So the recipient is, or the donor is blood group A. That means it is okay for this blood transfusion. But when we have crosses, that means this blood transfusion is not possible. This person is having blood group A, and the donor is blood group B. It is not going to be possible because agglutination will take place just as that. Then just look at this examine. This is when you say you see AB minus, this is the rhesus factor we are also going to discuss in this lesson. Still, don't worry. So look at the key. So the capital letters represent the antigens, as we've talked about. Small letters are the antibodies. X, that means not compatible, or the blood transfusion is not possible. Agglutination will occur. Compatibility is this mark, no agglutination. That is correct. And then we want to look at blood typing that assist during this blood transfusion process. So a person of blood group O can donate blood to a person of any other blood group. Reason, this is because they have no antigens but contains antibodies A and B. Such a person is called a universal donor. These people give out blood to all the blood groups. A person of blood group AB can receive blood from any other blood group because of this reason they have no antibody. This person is called universal recipient because they are receiving blood from any blood group. A person of blood group A can only donate blood to another person of with blood group A, or a person with blood group AB. A person of blood group B can only donate blood to somebody with blood group B or blood group AB. A person with blood group AB can only donate blood to a person with blood group AB. So remember, always blood screening has been very important step in controlling HIV. That blood screening means blood is examined in hospitals to ensure there are no dangerous diseases like the HIV virus and even some STIs like syphilis, yeah, even the hepatitis B is very serious. These are diseases transmitted in blood. It is therefore important to properly screen blood before transfusion is done. In addition to antigens A and B, there is another antigen on the red blood cell known as Rhesus antigen or antigen type D. This Rhesus antigen or Rhesus factor is present in individuals with Rhesus antigen in their red blood cells. Such individuals who have this antigen or Rhesus antigen or antigen type D are said to be Rhesus positive. Those lacking are said to be Rhesus negative, and these are the symbols used to denote Rhesus positive and this one for Rhesus negative. If blood from a Rhesus positive individual is introduced or transferred into a person who is Rhesus negative, this person who is having the positive or who is Rhesus positive develops antibodies against the Rhesus factor. Or we are saying if blood from an individual who is a Rhesus positive is transferred to somebody who is not having the Rhesus factor, this person who is a Rhesus negative develops antibodies or immunity, or the body responds against this Rhesus factor or the Rhesus positive factor, because the body of this person who is Rhesus negative is viewing it as an foreign material or as an antigen. There may not be any reaction after this transfusion. It might not have any effect, but subsequent or the next blood transfusion with Rhesus positive causes the blood causes a severe reaction and agglutination occurs. That is, the red blood cells join together such that there is no blood flow. That is likely to cause death. So we have a condition called erythroblastosis fetalis, that is hemolytic disease of the newborn, which results when a Rhesus negative mother carries a Rhesus positive fetus. That is when the father is Rhesus positive. We shall discuss in Form 4 in detail in the chapter of genetics how this Rhesus factor is inherited. But just know the mother will the gametes will fuse. So one from the mother and then half from the mother and then half from the father. And this Rhesus positive is dominant. That is why you will find the child is Rhesus positive and not Rhesus negative. During the later stage of pregnancy, the fragments or small portions of the Rhesus positive red blood cells of the fetus enters the mother's circulation. That causes the mother to produce Rhesus antibodies, which can pass across the placenta to the fetus and destroy the fetal red blood cells. Remember that at that time the fetus is still delicate, so its red blood cells can be destroyed. So during the first pregnancy, sufficient antibodies are not formed to affect the fetus. But the following pregnancies result in rapid production of Rhesus antibodies by the mother. This destroys the red blood cells of the fetus that now the mother is carrying. This condition is called hemolytic disease of the newborn, because that the mother is Rhesus negative and the Rhesus factor has been introduced. So this Rhesus factor, the mother's body will view it as foreign, but the first pregnancy will have no serious impact, but the second will have impact because its body will have developed antibodies to resist the Rhesus. because the father is rhesus positive. So the red blood cells of the fetus are destroyed, and that baby is not likely to survive when the red blood cells are destroyed. Most of the red blood cells are destroyed, and if the baby is born, it is anemic or having shortage of blood because the red blood cells have been destroyed, and then also the baby is having yellow eyes. This condition can be corrected using or by complete replacement of baby's blood with safe, healthy blood when still in uterus. The hemolytic disease of the newborn can also be prevented by injecting the mother with anti-rhesus globulin during the 28th day of the pregnancy. The mother's immune system will not recognize rhesus antigen as foreign because the anti-rhesus factor is already in the mother, so there will be no reaction from then. So thank you for being attentive. This is an assignment for you up to this part. Goodbye, enjoy yourself, and know your blood group. It is very important, and even the rhesus factor.